Good afternoon. Thank you so much. It's wonderful to be here and see you all. So this, what I would do to begin with is that I will try to place the conversation in the context and try to get you on board of the kind of questions we want to use or we have been using microscopes for. Okay. So I'll start off with Darwin's iconic quote, which was sort of relayed to you a couple of weeks back by Sir Paul Nurse. Right, so where he wondered about the endless forms of beautiful forms of life which are you seen on Earth. But what I'm going to do is I'm going to sort of deviate away from that question, and I'm going to bring you to another wonderful question, which is that I want to focus on or drive your attention to how do individual life forms achieve the same form and shape every single time, endless times. Right? So all of us, there is a diversity of shapes, but there's also an accuracy and fidelity in each and every organism that achieves that form and shape. So what you're seeing here on your screen is the development of 3 organisms, right, from embryo all the way till its adult form. And you can watch hundreds and thousands and millions of these processes in different embryos. You would get the same shape at the same time, and it's sort of, there's no difference in the process at all. Okay, so how do these cells or organisms achieve this accuracy and fidelity? So that's a question that is sort of driving developmental biologists, because any deviation, from this form or shape is going to cause developmental defects and diseases. Okay, so that's the context in which our conversation is going to sort of proceed, and that is what got me or many of us interested in biology in the first place. Okay, so what we will do is that to study this here in Ashoka, my lab uses a very simple organism. We call it C. elegans. Okay, and it is just a millimeter long. And so how many of you have seen worms? Worms, any worm? How long is that generally? What's 1 to 2 inches? So this worm is just a millimeter. Right? So you can hardly see it under with your eyes. But then with a microscope, you can actually sort of resolve the internal structures there. Okay, so why is that we use a worm to study these questions? So that's a question all of you should have, and we'll try to answer those questions going forward.
So, Hari, I just wanted to ask you a little bit about, when he talks about studying the worm, and then what is that technology which is there, which actually helps you see the worm? So do you want to get a little bit detail into that? Like, how do you even look at the worm? You can't clearly do any study by just looking at that image.
Yeah, so first of all, let me just say it's a real pleasure to be here. I mean, I know the time and effort involved in bringing people to this program, and I really appreciate it. And I appreciate your time as well. And also, I'm very inspired to see all of you guys here, because you are sort of the next generation of scientists, engineers, mathematicians. Yeah. Okay, so then to answer your question, these worms that Anup is talking about, you can actually see them with a naked eye, yeah. Right? So they're about the size of a comma in a sentence. The problem is if you want to see the individual cells, now you have to zoom in further. And so you need microscopes, which could be light microscopes of the kind that you guys may have used. If you want to zoom in even more, then you might use an electron microscope. But then the challenge is that the sample is dead, right, in an electron microscope. So some of my work involves taking even higher resolution images using light microscopy, and I know we're going to talk about that.
Yeah.
Yep.
Yeah. So I just want probably Anup to go to the next slide for them to just see the, why do you think there's 2 different colors there? Fluorescence. Okay. What lighting? Okay. I think there are some who knows that.
That's right. I mean, so one of the reasons why this organism is very popular to study development is because it is transparent, which means you don't need any special tool to study the process. As you can see all the internal organs, and you can also see those eggs, or what you call as embryos. And if you look carefully, as the embryo develops, you can actually see and measure things, right? And in fact, this was the first, this was the way, this was the approach it was taken to sort of find the lineage of the entire, all the cells inside the worm. However, given that recent developments in fluorescence, of course, also tools and techniques that allows us to see, visualize individual components inside the cell, as well as genome editing, like CRISPR, some of you are aware of it, CRISPR-mediated genome editing tools, has allowed us to sort of, tag or mark individual components, and then you can use various light wavelengths to look at these components inside at these worms. And what you're seeing here is actually a protein which has been marked with a fluorescent protein, which is a green, which is in color, which is a green fluorescent protein. And that you can that is sort of forced onto the cell membrane of all the cells in the worm. So what you see here is sort of a skeleton or a cartoon of the cell, but it's actually a live worm there. Yeah.
But then when you use this, lighting, as you call, which is the fluorescence, there are certain limitations which is coming up, and Anoop ran into problems.
Yeah.
And
So, but quite honestly, this is not how you would see it in a wild. In a wild, you won't see fluorescent worms. This is something which we create or we generate.
Yeah.
So maybe we should let you know where you find worms in the wild, right? So there is actually a high school project where all around the world, students go out in the wild, in the forest, searching for C. elegans, okay? And so C. elegans in the wild, you would generally find in rotting places, garbage dumps, rotting fruits, wherever, but you probably wouldn't find them in Sonipat for sure, right? So you need humid and damp and sort of more accommodative temperature. So then people have found, for example, C. elegans in Bangalore. So there are a couple of strains of C. elegans that has been isolated from Bangalore. So, yeah. So to answer your question, let me just again go back to our original problem. So before I tell you what the problems we face. So the question we ask generally in the field is that how does a one-cell embryo, which is sort of spherical in shape, get that tubular structure with all the organs and organ systems inside? Okay, so that's one question. And given that C. elegans is also present, this worm is also present in the natural habitat in the soil, it also naturally encounters a lot of bacteria and pathogens, right? So we also are trying to use this worm to study human pathogens and pathogen response behaviors. So again, it helps us that we can use these fluorescent tools to study. You can have a fluorescent bacteria and have a counter-colored fluorescent worm, and then ask the worm to sort of feed the worm with that fluorescent bacteria, and then you can see the bacteria inside the intestine. So these are, again, live worms with live bacteria, and then you can see how infection proceeds over time. But this is not just about studying biology. I mean, to study these or address these problems, we sort of use sort of diverse approaches, right? I mean, it's just not biology and the drawing of a cell and things like that. So there's physics involved, as you would see going forward. There is computation involved. There is a lot of biochemistry, pure chemistry, and also a lot of imagination as well you have to add into it. So that actually is what fuels a lot of answers, how we approach these questions. So maybe what I will go now is, to how do we get these images? So what are the kinds of experiments we do to get these things? So one important thing using microscopes, which we usually employ, is to get pretty pictures. But pretty pictures are not enough. Okay, we need to quantify them, because of course seeing is believing, but you need to measure to convince somebody. I mean, nobody would go by someone else's faith. So what you're going to see here is, this is an embryo on the surface of an embryo, okay, and it has just been fertilized, okay, and I'm going to play a movie, and I'll let you watch what is happening. So you can see a lot of things moving around, and on the left side of the image, you will see some particles which is sort of moving to this, which is moving to the left. Now it happens that these particles are actually protein molecules, okay? And these protein molecules are fundamental for the survival of the organism, okay? They are the proteins which actually hold the cells together. We call it cell adhesion proteins. Now, if you don't have those proteins, the cells would just fall apart. Now, the question is, we got a nice, pretty set of images, but what do we do with that? So what we want to do is we want to see how these proteins molecules move, how does it correlate with the cell division, and how can we correlate, if in the absence of those molecules, at what happens to the fate of these cells. So these, again, of course, you can manually sit and track individually, you can measure how fast it moves, but that's not the ideal way to do it. So here we write programs, codes, use computational power to analyze individual molecules, what we see, and then get an average velocity and then sort of plot it, okay? So this is an example I wanted to give you of how we use microscopy, computational power, as well as the genetic engineering that allowed us to get these fluorescent probes inside to address a cell biology question. Okay. Now, it's not that there are no problems. So when we do this, we always want to see more. Okay. For example, I would want to, at this resolution, I want to zoom in and see how many molecules. Is that cluster which you just saw? How many molecules are there? Although there is only one spot, are there 10 molecules, 20 molecules? Can I count the number of protein molecules, right? And I cannot just zoom in, because what I end up getting is just a set of blurry images. Okay, that's a serious problem which we face. That's one issue. The second issue which we have is that on the right side, on the bottom right, what you see is a live C. elegans, which is fed with a pathogen, and you can see infection. The pathogen is actually mounting an infection. Now, but if you notice, the images are very blurry, they're graining. So, and remember that these are, although they're 1 millimeter long, there's some thickness to this worm. And as you start doing microscopy of thicker samples, the quality of these images go down. And we are actually struggling to resolve or solve these problems. And I think maybe Hari could help us understand what the problem is and the possible answer for them.
Maybe I can ask, like, does anybody know what this image is? It's a protein, yeah. So it's a protein called green fluorescent protein, which was actually isolated from a jellyfish, and it glows green when you illuminate it with blue light. And so using genetic engineering, we can take this GFP and label more or less anything we'd like in a cell. So that's like a superpower, because we can light up anything. The other nice thing about this green fluorescent protein, or GFP, is that it's small. Okay, it's about a couple of nanometers on a side, so it's about the size of the kind of thing that we want to image, at the finest scale inside these worms. The problem is that when you image that GFP molecule in an optical microscope, what you see is something that is several hundred times larger. Okay, so that's called, the technical name for the image of one of these molecules, you might call it a point spread function. But it's basically, it's the impulse response of this microscope, if that terminology is something you've heard about before. So if we had just one molecule, if we were labeling just one thing, this wouldn't be a problem, right? Because any of you in this room, if I asked you to point out where the center of that GFP is, you do a pretty good job. Your eye would find a centroid, you'd find the mean position of that molecule, and you can do that with a precision much better than the width of the molecule. The problem is that in a cell, we typically don't just have one molecule, you have many, many molecules. And so in this bacterium, I've highlighted a couple of membrane proteins in green. And the problem is that if each of these is labeled with a fluorescent dye, and each of these fluorescent dyes looks much bigger than it actually is, now their images overlap. Their point spread functions overlap. And so this is like a huge problem in biological imaging. And it's a problem that has persisted ever since we've had fluorescence microscopes or even light microscopes. And so I was extremely fortunate in my career that when I was kind of finishing my PhD and wondering like what I should work on for my postdoc after my PhD, I ran into this guy, Eric Betzig. And so Betzig, in the 1990s, had this kind of theoretical idea for a way to get around this diffraction limit. So, if the problem is that you have many of these molecules and all of their images overlap, Betzig's idea was, well, what if you could isolate one molecule at a time and just image the emission from that one molecule? If you can find its central position well, then you can note that down with a computer, and then if you could turn that molecule off and turn on another molecule, by doing this kind of molecular imaging, essentially one molecule at a time, you could build up a much higher resolution image of the underlying sample. And this idea actually won him the Nobel Prize in 2014, and I was fortunate enough to work on this idea that won him the Nobel Prize. So again, the kind of key idea here is that if instead of imaging all the molecules at the same time, if you had some way of flipping on molecules one at a time somehow with a special kind of dye, then you can localize the center of each molecule and pin down its position with a precision much better than the width of that molecule. And so in time, you can kind of serially build up this super resolution image. So that's kind of the idea, which is in some ways amazingly simple idea. And just to kind of drive this concept home, I have a movie here of something that is hidden inside this movie. And in the top half of the movie, you can see the kind of image forming if you just add up all of the images of each molecule. But in the bottom image, you can see what happens if you can localize the position of each molecule as you build up the underlying image. And so you can see what's actually being formed is the name of this technique called photoactivated localization microscopy, or POM, right? So again, the sort of underlying idea is kind of hopefully clear, right? We're just isolating individual molecules one at a time, finding their mean position one at a time, and then showing you the image that results from each of these molecular localizations.
And sorry, that is computationally calculated, right?
That's right. So it's not enough to have the dye molecule or the microscope. You also need some knowledge of math and statistics in order to find the position of each of these molecules. So this would be sort of a good example of interdisciplinary science, right? It's sort of this marriage of different techniques that Betzig actually realized was needed in order to kind of actually put this method into practice.
Right. I also want to just put that word out to all the coding guys, the computational guys. It's not just about AI and robotics, okay? AI and robotics is good, but then there are many places where you can actually apply your knowledge to, make path-breaking discoveries. It would help biology, it would help physics, and I think it's truly, truly lateral and interdisciplinary.
Yeah, I think like one of the amazing things about this technique is that it's simple, and it could have been discovered years before it actually was discovered, because we knew about fluorescence imaging, we knew about single molecule imaging, we knew about centroid finding. So this is one of these ideas that it took the right kind of person to put these different concepts together, and I think there are many such ideas that are still available, right? So that's just something to consider. So my entry into this was, I was a little bit late to the party in the sense that I joined Betzig's lab after he had this idea, and it was my postdoc project to show this could be useful in practice. And so I spent a lot of time in his lab sort of imaging these cells that had these kind of special dyes that could be switched from an off state to an on state. In fact, I built a microscope to do this so-called super resolution imaging. And the kind of systems that I studied were called focal adhesions. So this is the kind of glue that sticks the cell down to a glass cover slip or onto another cell. And so in this image, you can see on one side of the image, kind of the conventional image of one of these focal adhesions. And then if you zoom in, you can kind of see this much higher resolution image, the so-called POM image. So if we zoom in to this box over here, you can see a little bit more detail compared to this grayscale image. But if we zoom in at one higher level of resolution, this difference in resolution becomes more manifest. And if I zoom in one more, now you can see what is just kind of like a diffraction limited mess, right? There's no detail in this image. It is resolved at the level of single molecules. And we're doing this in a light microscope, right? So it was this sort of discovery and implementation that won Betzig a share of the Chemistry Nobel Prize in 2014. So this was sort of what I worked on as a postdoc, but this is not the end of the story, right? So this is a fixed cell. The cell is dead. And one of the challenges in biology is that most things move in biology, okay? So these focal adhesions that stick the cell down, they move on the order of nanometers per second. And that length scale, that time scale is slow enough that we can actually knit together a live movie using this technique. And so in some of my work during a postdoc, I showed some of the first live movies at this resolution. But the problem is that many timescales in biology, including in these worms, occur at much faster timescales. So if you think about cell division or the movement of a molecular motor or calcium imaging or the movement of molecular motors at high speed, this occurs at much higher timescales. And in fact, this technique, this photoactivated localization microscopy technique is not fast enough to resolve these motions because of the time needed to flip these molecules on and off. And so I think there's still lots to do in microscopy. And part of the challenge is this, what I like to call this pyramid of frustration, right? So in microscopy, you're always trying to optimize one thing against something else. So if the cell is dead, it turns out using this technique, this kind of Nobel Prize winning technique, you can image at very, very high resolution using a light microscope if you have the right kind of dyes and the right math. But as soon as you have a living specimen, now there are all these trade-offs. So in order to image a specimen live, you typically need to shine a lot of light on the specimen to image at very high resolution. So, the higher the spatial resolution, the more light you need to shine on the specimen, the more easily you kill the sample. The higher the spatial resolution, the lower the temporal resolution, the lower the signal to noise, which is something you mentioned. So a lot of microscopy these days is in sort of about optimizing these trade-offs. And you mentioned AI.
Yeah.
So AI is one of the new tools that we have to try to mitigate some of these trade-offs. So I've kind of departed from this extreme kind of spatial resolution in my lab, and sort of a lot of the fun that I've had over the years is trying to image not just at very high spatial resolution, but also at very high temporal resolution. And so these are just a few more movies from my lab. So in the first movie in the top left, you can see a movie of these focal adhesions in a live cell. And that's great. You can sort of see at the molecular level how these adhesions are moving within a cell. In the sort of movie directly below it, we can actually image cancer molecules. So there's this molecule called HRAS, which is implicated in 30% of human cancers. And so we can image that HRAS at the base of a cell as it moves around inside a cell. In the middle movie there, you can see another movie of a cell now in 2 dimensions over time and in 3 dimensions. And this cell is another cell, and it's kind of ruffling. So it's peeling back from the cover slip. And we can image individual molecules of a molecule called alpha-actinin inside that cell. And then at somewhat larger length scales, I've also become interested in imaging cells inside these worm embryos. So the movie at sort of top right is a movie inside a moving worm embryo where we're looking at the cellular level. And the goal here was trying to track all of these cells as they move around, even as the animal is moving around. Yeah. And then at even larger length scales, we can image whole organs. And so this is, in the bottom right, is one more movie now of a whole mouse gut that we imaged at, now not at super resolution, but at sort of the cellular level. So this is just to give you an idea of the kinds of images that we can capture using a modern light microscope.
Right. And so, Hari, it's like very complex, right? You are actually using automation, you're understanding the physics bit, and then you have to understand the biological context. So, as you grew up, how did all these things fit into place? Because there is no course in any college which can actually offer all of them in one go. So how did you even think about taking this up?
Yeah, thanks. So maybe I can take a step back and tell you a bit about just sort of my own geographical journey, and then I'll tell you about my scientific journey. So like probably many of you, I was born in India. I was born in a tea plantation in Kerala. And my parents were actually the doctors for that tea plantation. So when they had me, we had to drive like 50 miles to another place so that I could be delivered. And I was in India for the first year of my life. And then my parents moved to England. And so I spent the first decade of my life in Birmingham in the 1980s. And actually England in that time was not a very fun place to be if you had brown skin. So I was bullied as a kid and beaten up and discriminated as a kid. And my parents actually were medical doctors, and they also kind of faced a ceiling in their work. We were living mostly in this kind of working class town. And so there was a lot of ill feeling towards immigrants. You know, there was a perception that immigrants were trying to take the jobs of the local Englishmen. And so we, for that reason, we moved away from England into the United States. So I spent the first year in the US in Cleveland, Ohio. And then the intention was always to move west because my parents, my mother had brothers who went to IITs who were among the first employees at Microsoft in Seattle. And so we moved west towards Seattle, where I went to college. And so I spent my teenage years growing up in the Seattle area. And then I moved to Berkeley, California, doing graduate school in biophysics, and then back east again as I had my own lab, and that's where I am right now in the Washington, D.C. area. Okay, so that's very long-winded history about how I grew up, but to get back to your question. So I was an undergraduate at University of Washington in Seattle, and I was a bioengineering undergraduate student. And the reason I chose bioengineering is because I couldn't make up my mind what to study. So I was interested in math and physics and biology and chemistry, and I couldn't make up my mind, and bioengineering was kind of like a grab bag major where you could take a bunch of classes in all of these different areas. So I pursued this kind of bioengineering undergraduate degree, but my real entry into scientific research came when I worked in somebody's lab. And so I worked in the lab of an analytical chemist called Jim Callis. And at the time that I joined Callis's lab, he was already kind of old, but he was already famous because he had developed a kind of pressure-sensitive paint. So this paint, if you illuminate it with UV light, it emitted a beautiful red phosphorescence. And that phosphorescence was quenched in the presence of oxygen. So the more oxygen pressure, the less phosphorescence. And what Callis realized is he could use this paint to coat the wing of an airfoil. So at the time that he developed this paint, if you wanted to measure the pressure distribution over a plane wing, there was no good way of doing it. You had to attach discrete sensors. And so the advantage of this paint is you could get this continuous map of the pressure surface of this wing. And so this was something that he became famous on. And so when I joined his lab, he had one last idea that he wanted to pursue before he retired. And so he had this kind of crazy idea. So if you think about how a honeybee flies, now switching topics, honeybees are too massive to take off by the laws of steady state aerodynamics. There's simply too much mass. And so at the time, this was not very well understood how they could fly. But Callis's idea was if you could take the wings of a honeybee and coat them with this pressure-sensitive paint, maybe you could directly image the vortices, the pressure at the surface of the wing. And so it was my task to take this pressure-sensitive paint and then figure out how to coat it on the wing and then image the kind of pressure distribution in a honeybee in hovering flight. And so as sort of a first step, it was unclear that we would have the signal. The honeybee beats its wings at something like 200 frames per second, 200 hertz. And so the first task was to figure out, did this paint have enough response where you could measure it? And so I actually built my first microscope. It wasn't really a microscope. It was really an instrument that we called the phosphorescence microphone. And so if you take this thin film of this paint and put it at one end of a PVC tube, at the other end of the tube, you could put a speaker. So as you probably know, right, sound is a pressure wave. And with a speaker, you can control the amplitude and the frequency of that wave. And so by varying the frequency of the speaker, we could sort of see, does the paint respond at 200 hertz? At the kind of one part in 10 to the fifth, I think on the order of a couple of pascals pressure amplitude that the wing was expected to beat. And so I built this sort of imaging system where I built this PVC tube with one end of the tube being a speaker, the other end being a thin film of the paint. And then I built an imaging rig where I could illuminate the paint as I fired these sound waves and then image the response onto a point detector called a photomultiplier tube. So I did all of that. And
How old were you then?
Okay, so I skipped high school, actually. So I was very young at that
So
time.
your age.
I was actually your age. And so here's actually a photograph of me when I was around your age, after I built this instrument. And I actually figured out a way of taking these honeybees, which we got from a local beekeeper. I put them in the freezer for 10 minutes to anesthetize the bees. I pulled them out, and then I quickly pipetted a thin film of this paint on the bee, which was still groggy. And so that wasn't enough. We also had to figure out a way of coating the wing of the bee with this porphyrin, right? And so the molecule itself wasn't soluble on the surface of the wing. So I had to test a bunch of polymers to figure out how to coat the wing of the bee. It turned out that the best polymer was beeswax, which was actually in hindsight not so surprising. So here you can see actually a wing of a bee glowing red, glowing phosphorescent, as we displace the oxygen with a nitrogen gun on the thin film on the wing of the bee. And so this was all very educational, but it was actually a failure. So even though I figured out how to coat the wing of the bee, we could never get the camera with good enough signal to noise ratio to image the bee in hovering flight, even though the bee could actually take off with this thin film of paint. So this was my introduction into instrumentation, and it was sort of inspiring because even though this project failed, there were a whole bunch of different things I had to learn, right? Some chemistry, some imaging, some data processing en route to this. And this sort of convinced me that this was something interesting and fun that I could do in graduate school.
So when you try and do these things and it fails, then how do you go to new ideas and how do you pick it up from there?
Right. So I think the key to remember in sort of any kind of scientific field is it's only a failure if you sort of stay down, right? So the key is to pick yourself up. And in science, sometimes you work on a project and you reach a point where you can't proceed any longer. So this was one of these examples. But fortunately, I had another chance. I went to graduate school. And in graduate school, I had the opportunity to kind of pursue building instruments in a totally different context. And so at Berkeley during this time, there was a lot of interest in a technology called optical trapping. So many of you have probably seen Star Trek. You might have heard about the technology of tractor beams. It turns out that you can make a kind of tractor beam at the nanoscopic level if you take an infrared laser and focus it down to a diffraction limited focus. That pulls in any object with a higher refractive index than the surrounding medium. So if you take a polystyrene bead, you can actually hold it in the focus of this so-called optical trap at the focus of a laser. And then you can use that trap to impart forces and to measure nanoscale displacements. So if you anchor at one end of the bead a biomolecule, and then you have that biomolecule tethered between one bead in the optical trap and another bead held in a pipette, now you can impart forces to the biomolecule by kind of moving the optical trap. And also if the molecule moves inside the trap, you can sense the force on that molecule by monitoring the displacement of the bead at the trap. So this is a very sensitive assay for measuring forces or displacements. And so at the time that I came to Berkeley, there were many people that were using this technology to measure forces on biomolecules in vitro. It turns out that if you want to do this in a cell, this fails because the laser beam burns the cells, and anything with a higher refractive index than the cytoplasm gets pulled into the trap. So you don't have any selectivity, any biological selectivity inside the cell. But I was really interested in sort of seeing, could I measure forces inside cells? So, getting back to the sort of C. elegans, it's not just important to measure where things are going. You would also like to be able to measure forces on these biomolecules as, say, an embryo develops. And so it was clear to me, as a graduate student, that this idea of measuring forces was sort of something that one couldn't do it very well inside a cell. It was also clear that this optical trapping technology was not the way to go. So this was sort of, no matter how carefully you worked at it, this would be very difficult. And so I had this kind of crazy idea, what if I could measure forces without the laser beam, without this optical trap? What if I could use the molecule itself as a force sensor? And so as a graduate student, I kind of developed, I think, the world's first molecular force sensor. So the idea is that a single molecule is a kind of a spring, okay? And if the spring is relaxed, it's floppy. So if you have two fluorescent dye molecules, when the spring is floppy, if you attach these dye molecules in the right way, the molecules are far apart and they can't communicate If you excite one, there's no energy transfer to another. But if the molecule I got that a little bit backwards. If the dyes are close together, if the molecule is rigid, one molecule can transfer energy to the other. But if you pull on the molecule so that you separate the dyes, now you don't have this kind of energy transfer. And so depending on the force state of the molecule, you can measure this kind of force optically, and you can take this force sensor and put it into, say, a curved DNA loop to measure forces. So this was an example of kind of a molecular force sensor. I had to build a microscope actually at the time.
So just to check you on that, so you can imagine development as a mass of cells, because cells keep dividing once a fertilization happens, and that mass of cells has to get into a particular shape, right? So which means the organism has to sculpt itself into a defined shape every single time. Where do forces, I mean, do you think, of course forces are involved in sculpting. Do you think one could measure those forces? Is it over time? Is it
Absolutely. So one application where this is very important would be in cell division,
right,
for example, right? So if you guys remember those images of the embryo is pulled apart as it's divided. The two cells kind of are forced apart. And so that would be one application where it would be very valuable to understand the forces and how, when embryos are not allowed to divide, how these forces go awry, so to speak.
And just to let everyone know, we are talking about piconewton scale forces. So that's what we are trying to measure here.
Right.
So, yeah.
So in this particular case, I had this idea for measuring forces that actually worked in principle, but I didn't understand anything about cells at that time. And so this was another example of a project where I was able to publish some papers. I showed that you could measure these piconewton scale forces using this kind of entropic spring. But I didn't know any cell biology, and so I didn't know about endonucleases, which chewed up the DNA. I didn't know that certain dyes couldn't cross the cell membrane. And so this was another failure, so to speak, at the time. It turned out that the idea was good. And so people later came along and took this idea and showed that with fluorescent proteins instead of synthetic dyes, you actually could use this kind of concept to measure forces inside a cell. But at the time, it was another failure. So maybe another lesson here would be, you can have an idea and develop it, and then, maybe the idea pauses for a while until somebody else brings up a particular technology that makes it feasible. This is another concept that happens in science over and over and over and over again.
And so now we are moving into a very interesting area, and also to check whether you are sleeping or not, we have a question for you. Does any one of you, they know what this is? Spectroscope, okay. Any other guesses? 3D printer, okay. Yeah? Okay, 2 spectroscopes, okay. Transmission electron microscope, okay. Okay, one more guess.
It's a spectroscope.
Okay, everyone is very sure it's a spectroscope. Okay, Hari, why don't you tell them what it is?
Right, so it's actually a microscope in my lab. So we use this to image cells and embryos. And the spectroscope is a good guess because some of you might have seen some lenses, some mirrors, some lasers, but this is actually what a modern microscope looks like under the hood. And it's interesting, right? Because it looks nothing like the thing that some of you guys may have used in the lab, which is the thing you're actually used to thinking about, right? With sort of an eyepiece, an objective lens, and a condenser lens. In fact, if we look more carefully at this thing that I showed you, it is a microscope, and there are objective lenses, but there are many objective lenses. There's not just one. They're arranged in unconventional ways. So yeah, often even the kind of simple microscope that you're used to seeing, it starts out looking like the thing that is in the lab, right? And then it gets packaged in a way where it's very difficult to kind of actually see how it works or what's under the hood, so to speak.
Okay, so I think also the thing is when you talk about microscope, for many of the students, it's just a compound microscope where they're looking at maybe red blood cells and onion peel, but it's much complex than that. And I also noticed that, when we floated around the form for picking up the course here, for microscopy, they see only biology related to microscopy. They don't see the physics side of it, which is all about physics and optics, because they will all go for the physics students who are interested in it, or would all go for the physics course, but they don't see microscopy as physics. So maybe, Hari, if you could just let them know about the kind of physics that's involved in it, I think that'll also be great.
Sure. I mean, so maybe I'll give one example having to do with imaging things deep inside a specimen, just to give you some sense of the physics here.
Yeah.
So this falls under a rubric called adaptive optics, which you may or may not have heard of, but the intuition is kind of like this. So suppose you have like a sample, like an embryo, this is this egg-like shape here. If the imaging is ideal, every point on that embryo is a source of spherical waves in an ideal world. And if the imaging system is ideal, that microscope converts those spherical waves into plane waves with a flat phase. So if you were to be able to sort of put a little sensor inside the microscope, in the ideal situation, the phase of the wavefront is flat.
Yeah.
Okay, in principle. But in reality, life is never that good, and the rays bend due to variations in refractive index in the sample due to imperfections in the microscope. So another way of saying that is that the light doesn't all get to the same point in the sample, and the light doesn't get to all the same point in the detector in the microscope. And again, if we think about the phase, the wavefront in the microscope, there are peaks and valleys. And these peaks and valleys mean that your imaging is never perfect in a thick sample. Even in a thin sample, it's never perfect, but especially as you image deeper and deeper, this is sort of a problem. Now, in response to this problem, there's a field of imaging called adaptive optics. And the intuition behind adaptive optics is actually very easy to grasp. The idea is if we could measure that aberrated wavefront, that's called wavefront sensing. If we could apply an equal and opposite wavefront, then we could cancel that aberration and we could restore a flat wavefront. And it's as if the aberration never existed in the first place. It's as if the rays all came from single points forming spherical waves. And again, the microscope produces plane waves and the imaging is perfect. And this idea of adaptive optics is really important across imaging, okay, not just in microscopy. So, we can think about astronomy. And in astronomy, if you try to take an image of a planet in a ground-based telescope, you can kind of see the planet. I mean, you can distinguish, the ring in the planet from the body of the planet, but it's kind of fuzzy, right? And the reason is the variations in refractive index that kind of are between the light from the planet and the imaging system that kind of screw up the waves on their way to the detector. And the astronomers, because of adaptive optics, they've invented ways of getting around this problem, where the light from the telescope is kind of siphoned off to something called a wavefront sensor, which tells the astronomer how the waves are out of phase. If they can understand that, then they can apply this equal and opposite wavefront. So to collect a flat wavefront again, as the telescope is imaging the object. And this is enabling in astronomy. So with the adaptive optics on, you can see a lot more detail in the planet, right? You can resolve the ring structure, some dust clouds near the ring, the moon is better resolved, and so on. So you can see more if you can cancel these kind of imperfections in the imaging system. Now, this was an example in astronomy. The same thing works sort of billions and billion times smaller in microscopy. So here's an image that we collected in our lab of a single cell. And there's something in this cell, which is a molecule called myosin. The myosin is important in single cells and in worms to move things around, to apply controlled forces. But if you image this cell just a few cell layers into, say, an embryo, it becomes very difficult to see the fine structure in that cell because of these aberrations, because of these distortions in the tissue. And so with adaptive optics, after turning the adaptive optics on, we can restore the image, right? So it's like night and day. Now you can see these myosin doublets that are actually there inside the cell. And so again, there's a lot that can be done in the field of cell biology by using these techniques, much like astronomy. And there's still a lot of room in this field. If one can find better ways of wavefront sensing, better mathematical algorithms, better computational algorithms, you can do this more efficiently and faster.
Right. So then, I mean, most of the things and techniques that you are designing or you are inventing, it can be applied to other fields as well, right? So how does it work? So when you design something, do you always take into account as well other fields as well, other than biology, that, you know, it should be useful there? How does your design come into play there?
Right. So, yeah, the short answer is yes. You know, breakthroughs in fields often happen when you can take an insight from one field and apply it to another. So I'm always on the lookout for innovations in new fields. I mean, one of the latest advances, of course, is in artificial intelligence. But there are also these old school ideas from the 1980s, you know, where the astronomers worked out ways of accounting for aberrated wavefronts that are applicable to microscopy, and nobody ever looked carefully at these things. So this is just one example, but there are many opportunities like that in science. Now, this isn't the only way that we look for solutions. So sometimes a biologist comes to me and, you know, they want to image something at higher resolution or at higher speed, and they have a very specific problem. So that also is how it works, right? It's not only you look.
Yes, it's a two-way process.
Exactly.
Yeah. Okay.
I think the key is to kind of always keep your ear to the ground for interesting advances in other fields, right? And think about how they might apply. And many of them won't, but some of them will. And sometimes, again, that's where the breakthroughs happen.
Right. And so now we have seen a spectrum of your work ranging from astronomy to microscopy, and having all these, developing all these techniques and working with a number of people all across the world. But it looks all hunky-dory right now. So I just want to know, what was the behind the scenes?
Right. So one thing to remember, I think, as you listen to all of these talks, as you listen to these lectures, is that you're really only seeing the tip of the iceberg, right? I mean, I'm giving you a sort of nice, whitewashed picture of what actually is happening. I'm just showing you the successes. I've tried to give you some sense of some of the failures along the way, but really the failures are the meat of it, right? So the publications, the talks that you see are the tip of the iceberg, the shadow of the science. The underlying thing, the real meat of what it's like to work in a lab, are the failures, the disappointments along the way. And you only get a sense of that really when you actually work in a lab, when you work in kind of a scientific research environment. And I would say the essential thing is to not get discouraged, right? So, if there are successes that we've had, it's only because we have failed more often than others and learned from the success. So that's sort of the key thing, I think, no matter which field you're in, right? I mean, there will be obstacles in your thinking, there will be problems, overcome them, right? And this takes time and persistence.
Right. And if I ask you about, what makes it to have a good project, how do you know that this is the project, this is the line of thought I should have, and this is where I should go?
Right. So, I mean, I like to kind of think a little bit abstractly about that. So, one of my scientific heroes is this biologist, Jeremy Nathans, who discovered the genes for vision as a graduate student. And he likes to talk about this kind of Venn diagram of projects, right? So it's good to work on important problems. That's how you can have an impact. But it's also good to think about, there are many important problems where you don't have a line of attack, right? Yeah. I mean, time travel is one, teleportation is another one. If you figure out how to do that, that's like an instant Nobel Prize, right? But we don't have ways of attacking these problems that are practical. Right. So it's also good to think about feasibility, but also it's good to work on problems that are not obvious, or at least the solutions are not obvious. And the sweet spot is kind of the intersection of these areas, right? And I'm a tool builder. So, the way that I think about problems is I think naturally about failure and what makes an imaging tool not work. And so what I found, at least in my career, is that if you can make the doability circle bigger, if you have some idea that makes some problem easier to attack, then very often that makes the problem feasible, and that can lead to success.
Right. And do you think that, because many of them are looking forward to having a very, very fruitful career, so is there something that you would like to tell these students who are here, who are listening to you, about, is there any mantra for success, or how do you gauge success that way?
Right. So, I mean, there's no mantra, but like one thing that I think about are sort of the factors that are in your control versus the factors that are not, right? So you all wouldn't be here if you didn't work hard, if you weren't interested in science. And there's a sort of hierarchy of control, right? So the thing you can control is how hard you work, how hard you study. There's this saying, no pain, no gain, and that's certainly true in science, right? If you want a career in academic science, it's very difficult to do that on the 9 to 5. Of course, it's also important to hone your abilities, to be smart. But again, all of you have that in spades, right? And your intelligence can be honed. You have some native degree of intelligence that can be honed by hanging out with people that are smarter than you are, that can think more clearly than you can. And I always get very nervous if I'm the smartest person in the room. Luckily, that's not the case here. But I'm often the hardest working person in the room, and that is something that I can control. The thing that you can't control is luck, right? So you don't pick your genes, you don't pick your environment, your educational background, your parents. But again, there's some truth to this idea of luck favoring the prepared mind, I think, which is something that Pasteur, it's attributed to Pasteur. And so what I would say is to write down your ideas, right? Because many times you might have an idea and then you figure out after the fact somebody else had the idea, but sometimes not. And those ideas may not be practical to work on at this point in time, but you might learn something later on, or there might be a piece of technology that makes your idea workable. This has happened to me many times in my career. So maybe that's the advice that I would give. Continue to think creatively, write down your ideas, and come back to them periodically.
No, that's fantastic. So one more thing somebody told me many years ago when I was just starting up in my undergrad is that to succeed you need to fail often and fail early. So you don't get hurt. You know how to get up and bounce back. So I think that's something which I would want to share.
No,
Yeah.
I think these are both very important points, and these are things I hope that you would just not, throw it away in Takshila and will take it home. So thank you, Hari, and thank you, Anup, so much for sharing your work
Thank you.
and life, and also all these aspects of, how to get your work done, how to become successful, and, and how to deal with your failure more than anything. So with that, we come to the end of this panel. So I guess we are open for questions now. Okay. I'll just try a new person, maybe here.
Thank you. Hi, sir. So I wanted to ask, how exactly do you switch on and off the GFP molecules? And then, how do you use computation and AI to mitigate the trade-offs, as you were saying, while imaging?
Yeah, that's a great question. So if you look at a normal GFP molecule, it's not easy to switch it on and off. If you excite it by shining blue light, all of the molecules come on. But there is a class of molecules which can be, they have basically two states, a dark state and a bright state. And by cycling them with a different wavelength of light, you can engineer the imaging such that only a few molecules are on just by chance. The majority of them are in a dark state, and just a few of them can be excited. And just by chance, those few are well separated with respect to their diffraction limit. So it requires a special kind of molecule. And then, sorry, your second question again, can you say that again?
Yeah, I was asking, how do you use computation and AI after that to mitigate the trade-offs, as you were saying?
Right, so in the case of the switching, for example, we use computation. We take each image of a molecule and we might fit a Gaussian to find the center position of that molecule. This would be one example of how we use computation. In another example, we would use artificial intelligence to denoise our images, right? So then we can turn the laser down an image for longer and keep the cells alive for longer. That would be another example.
Okay, thank you.
Yep.
My question was that image of the microscope you had shown us, was it a compound microscope or something? And my second question is that, the movies you made of the microscopic creatures, is there a certain procedure or it can differ from creature to creature?
Right, so that microscope was not a compound microscope, it was a fluorescence microscope, and it was actually one of these very high resolution microscopes that we call super resolution microscopes. So it was something that we hope might one day be commercialized. But when it gets commercialized, it becomes more usable and, in my opinion, much less interesting to look at because everything is then boxed up and it's hard to figure out how it works. And then, okay, your other question was about kind of sample preparation and how do we prepare the samples. So there's no formula for that. So we would prepare worm embryos differently than cell culture, different than mouse tissue. So there are established procedures, but they differ from sample to sample.
You talked about that paint that was pressure sensitive and was used in airplanes to measure the pressure and vortices. What are the other fields that you think the paint could be used in?
what are other applications the paint can
Yeah.
be used in, right? So as far as I know, Boeing is still using this paint to measure air pressure. Another one is in cells, there's a lot of interest in measuring metabolic reactions. So you want to know how much oxygen, how much pressure there is inside a cell. And so there are variations of this paint that people have actually applied to tissue culture in order to measure kind of in bulk the kind of oxygen that is present within or nearby cells. So this would be another application of that paint.
So, sir, it was very interesting lecture, and my question is related to the development in the worm.
Yeah.
How do you study the development in the worm using this microscope? Because in each different organism, the development takes place in a different manner, and you will have to use different frequencies to find out the movement of the embryonic development. So to find that out, what are the procedures you follow?
Right. So as far as like what kind of microscope we use to image the worms, in our case, in my lab, we use a microscope called a light sheet microscope. And the idea, the intuition there is, if this is the sample, my fist, in most microscopes, you shine light everywhere through the sample. And that's very damaging to the sample, or it can be. You can bleach the fluorescence, you can damage the specimen. So instead, we make use of a very simple idea, which is that we focus our laser beam into a thin sheet that only illuminates the focal plane. And then we sweep that light sheet through the sample at high speed. And this really cuts down on the amount of light that the embryo sees, because we're very interested in my lab at imaging at very high speed. So that kind of touches on the second part of your question, which is how do you decide what frequency with which you're imaging the specimen? And the answer to that question is it really depends on what you're studying, right? So if you're studying something that occurs on a slow time scale, like morphogenesis, you might use a different kind of microscope where you don't care so much about imaging at very, very high speed. If you're imaging sort of cell division and you want to resolve that sort of at a very high speed, then this would require a different microscope. So I think the answer to your question in some sense is application dependent. I don't know if you have anything you wanted to add to that, Anup.
No, I think that you covered it.
How can we use microscopy not to just identify physical diseases, but also mental illnesses or mental diseases?
It's a great question. Yeah. So one of the reasons I like this worm is that in the 1980s, somebody studied the worm, a guy called John Solston, with a very simple microscope, actually a compound-like microscope, like the ones you may have used. And he was just looking by eye. And one of the things he noticed is that certain cells in the worm embryo were always programmed to die. This is called apoptosis. And it turns out that every animal has apoptosis. And this is called programmed cell death. It turns out that cancer is a disease where you don't have enough programmed cell death, or the genes that underlie cell death aren't functioning. On the other hand, neurodegeneration in the brain is sometimes a disease of too much cell death, right? And so by studying the genes that underlie cell death in the worm, which were discovered in the embryo, it turned out those same genes underlie what happens in the human, like a disease state. So I think one answer to your question is these model organisms are still very powerful today because you can study them sort of in entirety. You can look at the entire worm embryo. You can look at that worm embryo as it develops from a zygote to a little worm, and you can study disease, things that are causative of disease in a way that are not feasible in a human. And with all of these modern techniques like genomics, you can then relate these things you see in the microscope to the genes that underlie human disease. So it's actually an extremely exciting time to use model organisms to uncover principles of disease at the basic level and then transport them all the way up the food chain to us. There's never been a better time to use microscopy to study disease. And the reason is these diseases are conserved. The worm, I think, has something like 70% of gene homology to the human, disease genes that are conserved in the human, even though the worm looks nothing like a human.
So just to add one point, I think some of your batch mates are actually doing exactly the same thing. They're looking at a gene that essentially causes nerve diseases in humans, and they're looking at it in the worm. So yeah, actually what he just said, it's being done as part of your program.
And another question is, it's a short one. How can we use force sensors to train or can be used in prosthetics or robotics to give more sensitivity to touch?
Yeah, I think that's an interesting idea. I would not use the kind of force sensor that I discussed, but I think that is certainly important, right? If you're trying to design some robotic system to pick something up, I mean, of course, it's going to be important to measure the tactile forces, right? And so one would need to know, what are the forces involved as you touch something, but one would not use probably these molecular force sensors.
Okay, thank you.
Thank you.
Hi, professors. So I'm an undergrad here. I'm doing math, and I just had a question. At your level, as in now, you guys are researchers, right? And I think Janelia is that the name of the research institute. So, in your experience, are academics still operating in silos? Because at least in my very limited experience, the mathematicians aren't talking to physicists aren't talking to the biologists aren't talking to chemists,
Right.
and so on and so forth. However, when I was looking at your guys' profile, I couldn't really pin you down to, okay, he's a physicist. No, but he's an engineer. No, he's a biologist. So you're kind of doing everything and combining them to get insights.
Right.
So in your experience, is it getting better? Are academics talking amongst each other, or is it, just plateauing?
Yeah, I think there are institutions like the one that I work at that prize that kind of interdisciplinary work, right? Where, yeah, exactly as you say, it's very hard to pin down one scientist as one thing. And that's by design, because we like to talk to each other and work on problems. I think that's really important, and I think it's good to find places that foster that. But I also want to say that I also think it's important, if you're passionate about math, study pure math. That's also important. Study physical mechanisms if that's what floats your boat. I mean, to some extent, this is a matter of taste, and everybody is different, right? So I don't have any. I would never say to somebody, you should only think about things in an interdisciplinary way. I mean, oftentimes that is how breakthroughs happen, but we also need breakthroughs to happen in so-called pure fields as well. So I think there's room for all of us. Yeah.
How did you manage to get a chance to research at such a young age?
Right. Okay. So that was, again, pure luck. So I was in middle school in the Seattle area, and we opened up a newsletter one day, and the newsletter advertised this program at University of Washington where they took 13 and 14-year-olds and then put them in this special program where they prepared us for college in 1 year. And so I was bored in school, and I was like, this is the thing for me. I have to do this. And it's not for everybody, but it was the right program for me and my brother. So I was fortunate enough that this program existed at University of Washington, and I was also fortunate that I learned about it just randomly. So that's kind of how that happened.
Yeah, so you mentioned how computational biology and computational mathematic modeling works with all these different fields in biology. And so I was kind of wondering, how does computational biology and experimental data work with each other? And what are the key breakthroughs that computational biology is helping further when it comes to experimental data? Right.
So I guess a big recent one would be AlphaFold, right? So protein design was kind of stymied for decades, right? I mean, this protein folding problem of how you predict the 3D structure from a sequence was kind of an unsolved problem. And this was a problem that lent itself very well to computational techniques, just sheer amounts of data, which also existed, right? We knew we had the protein structures. And so it just took sort of the realization that the data existed, and then the neural network models were sort of existed, and this was a good match. So this was a case, I think, where smart people realized that there was this interesting problem that could be posed in this input-output way, which is very amenable to AI, and the data, high-quality data, also existed. So this would be one example of a recent breakthrough. But maybe an older example would be, the genome was sequenced, right? And so you have all of this information in terms of DNA sequences, but how do you make sense of that? How does this actually, what are the patterns of overlapping gene expression? What are the genes that cluster together? This was a problem for bioinformaticians, right? So these were computer programmers that could align sequences and then talk to the biologist and then learn, okay, if these sequences were aligned, what did this mean for the organism? So in both cases, I guess, intersection of computer scientists with kind of domain experts, with biologists that could say, you guys should study this thing, or we need a tool that aligns these sequences. So that's maybe another example of communication, right? Where there had to be communication between these two disparate groups that didn't have the skill sets individually to kind of make progress.
So my question is, how is phosphorescence microscopy different from fluorescent microscopy, and in terms of its application and uses? And my follow-up question is, how can different slow chemical processes like metabolism be studied using them?
It's a great question. Yeah. So in fluorescence, you excite an electron, right? And that happens and then the electron gives off a photon as it decays to the ground state. And this happens at sort of the nanosecond level typically, because of the kind of nature of the chemistry that's involved. In phosphorescence, you're typically coming down from a triplet state, and this has a longer lifetime. So the main benefit, drawback, depending on how you look at it, is the time scale involved. So phosphorescence is a longer process, which means that it's very useful for probing longer scale processes than fluorescence. That's the key difference. It's a question of time scale. So in some applications, the biology, for example, occurs on a fast time scale, and you want a fluorescence sensor. On other cases, the biology occurs too slowly for a fluorescence sensor, and so you might want a phosphorescence sensor. And so it really is kind of application dependent. And so that's sort of also, in a way, the answer to your second question, which is that depending on, so metabolism, some things are fast, some things are slow. And this is an area of active development, right? You would want to develop sensors that are either phosphorescent or fluorescent, depending on the kind of use case.
Hello, sir. I'm Shravya. So I had a question about what are your views on AI, on the future of AI in microscopic imaging, and how would you feel or have a perspective about a project which uses AI and microscopic images to detect certain diseases such as sickle cell anemia?
So I think AI is an incredibly powerful tool in microscopy because you can learn things statistically from data. And as microscopists, it's very easy to acquire lots and lots of data. So you can make these powerful statistical inferences, and we use this in my group to denoise data, to turn the laser down, as I kind of previously mentioned. Now, there's also a danger, which is that these AI models, very often they're applied without physical principles. They're simply learning input-output relationships from data. And so if you're looking for like a rare state, these AI models can be biased away from the rare states. They simply learn what there is in the data. And so exactly as you're saying, if you want to look for something that is a little bit outside the norm, the AI models can fail or they give you garbage. So that's the cautionary note. I think like one future application is to teach the AI models to harness the uncertainty, to harness the predictions that don't make sense, to look for these kind of unknown states. That would be one future application.
If we want to be successful in our careers, is there like an ideal age at which we should know like what we want to do? Like lots of people still have no clue about what they're going to do in the future, but some people already have it decided. So like what's the advantage of knowing it before, and like what's the best age to know it at?
Yeah, it's a great question. I think the answer is there is no best age, for any of this stuff, in my opinion. So I kind of stumbled into each of the things that I told you about. I lucked out by going to this program, this early entrance program. Somebody told me that it might be a good idea to work in a lab. So I talked to a professor after I heard his lecture, and then I went up to, and then he said, I have an opening. I was probably the last person in my class to learn about graduate school. I kind of learned about this as a junior, relatively late. And so then I was like, okay, maybe this is a good idea. I went to graduate school. I didn't meet Betzig until I was almost done writing my thesis. I didn't want to do a traditional postdoc. Many of my friends were going to well-known labs, and that just struck me as totally boring. So I just interviewed with this one guy, and he was an unknown at the time. He was out of science. So my career is totally serendipitous, and I could only connect the dots in hindsight. Now, that's just me. You might look at another field like mathematics, where some of you probably have prodigious talents in math, and you like to do that. So that's great. I would say pursue those talents. But for those of us that don't know, there's absolutely nothing wrong with not knowing. I would say follow your interest and head for the murky water, right? So don't do what the other people are doing. find your own path. That's maybe the best advice that I could give you. And there's no shame in not knowing at the college level or at the graduate student level what you want to do. Keep looking until you find something that you like to do, that you're willing to invest your hours in, right? Because whatever you do, if you want to make an impact, it will take lots of time. It will take lots of time. And there's no sense in doing something that you don't enjoy. That's what I would say. Fly like you, not like them.
Good afternoon, sir. My name is Pavitra. I have heard about this technique known as dual view selective plane illumination microscopy, and I suppose it was developed at the NIH. It enables rapid high resolution live imaging of living cells with minimal phototoxicity. So apart from this, are there any other such techniques which enable the same level of microscopy?
Yeah. Thanks for the question. So it's probably no accident you mentioned that. I invented that technique, and the intuition there is you look at a sample from multiple directions and you gain more information about the sample. So in the case of these embryos, what typically happens is your ability to look in Z is much worse than in XY. But if you just look at the sample from a different perspective, you can gain that information back. Another way of getting the similar resolution is to make your light sheet very, very thin. And there are other people, including Betzig, that came up with methods for doing that. So there are often many ways in microscopy of getting at the same thing, and they have advantages and disadvantages. Yeah.
During the lecture, you talked about how you didn't really want to choose between biology and maths, so you chose to become a bioengineer. So could you just explain like what are the demands for that field of science? Like what all is expected, and like how challenging could it be to become like that sort of scientist?
Yeah, so I think there's no right or wrong answer to your question. It depends on where you do a bioengineering degree. But for me, something that was attractive is I could take a lot of courses in biochemistry and in thermodynamics and in math and combine these things into a degree. And that's just the way that my mind works. Now, the disadvantage of that approach is that I couldn't go very deep into, say, physics, right? So a physicist could learn a lot more at that time about sort of high energy physics, let's say, than I could, or electromagnetism. And so I actually later in graduate school, I became more interested in physics, and I took some classes at Berkeley in physics. So the requirements at the time that I took it were kind of like, sort of like broad, right? Like you needed to take some chemistry, some biochemistry, broad, but not very deep to graduate. And that sort of appealed to me at the time because I couldn't make up my mind. I guess the counter argument might be that if you want to go deep at the undergraduate level, if that's sort of what you're interested in, then I would advise picking a different degree. And again, the door isn't closed after your undergraduate degree. So there are plenty of physicists, people that have a physics degree that later find biology, and later, they kind of want to work in that area. And one of the wonderful things about science is that the door is never closed. I mean, there's nobody saying to you you have to stop at this phase, right? You might have to invest more time than your peers if you want to switch field, but you can always do that. And I've done that many times in my career.